The SI session, which is recorded, so I'll post it on Canvas as well. This thing I talked about on Monday. So you may be given a question where the way they phrase it is that there's this brand new peptide that's 10 amino acids long. And they perform an experiment where they use some enzyme and it ends up cutting it into two pieces. Don't care about that enzyme. All its job is to cut this brand new amino acid sequence into two pieces. Then they start over. They do the experiment again with the same 10 amino acid long peptide, and they use a different enzyme this time. And this different enzyme decides to cut it into three pieces this time. Only from these two experiments, you're tasked with trying to figure out what the original DNA sequence was. Okay, and here's how you do it. This first statement here gives you two fragments. You don't know which one comes first or not. So the quick way of solving this is to write the two possible answers that you can get. Right? The two possible answers would be where that's first, and this comes after it. And the other answer will be, let me make some room. Where this one comes first, followed by this piece. Okay? These are your two possible answers because those are the only two fragments you made in the first experiment. You now use the information in the second experiment to figure out which of these two is the right answer. This second experiment has different fragments, but the amino acids are next to the same amino acid that they were in the first experiment, meaning this phenylalanine is always going to be next to a methionine and a serine. So I'm going to look down here to figure out which of these answer choices satisfies that. Unfortunately, this one has a phenylalanine next to a serine and a methionine, and so does this one. Well, so that's not enough information. Anyone want to take a look, after looking at these fragments here, which of these two choices down here must be the correct answer? The first one or the second one? The second one, and the answer is you go look at more of these fragments and you find out which one is right. This one here says aspartic acid, tryptophan, and lysine. Nope, that's not a good one, excuse me. Let's see, what's a good one? Leucine and serine? Yeah. Nope, not a good one either. I am not good at picking these. Basically, just pick any of these fragments and check your answer to see if you find that fragment there. So I'm going to highlight this and I'm going to look for it down here. Serine, phenylalanine, methionine, and leucine, and arginine. So that's correct. They're right next to each other. How about aspartic acid, tryptophan, and lysine? Those are right next to each other. And finally, leucine and serine. Those are right next to each other. If you do the same thing for the first sequence, it's not going to work. Okay? Yes?
Will you give us some of that?
Yeah, you will be given a question that's like...
Maybe just like easy?
Yeah, a lot of people don't find this easy. I will tell you that. A lot of people don't.
Or is that...
What's that? Yeah.
Would this be for like a test question or just credit?
This would be a test question because it's multiple choice answers for this one.
Okay, the only one, and that's guaranteed an extra credit?
The extra credit one will be the sequencing one. That's guaranteed to be the style of question that you would get. It'll just be different clues. Yes. The extra credit question has a maximum of 5 points of extra credit, 5 percentage points of extra credit. 1 second, you had a question? Yes.
So we're past chapter 4 at this point. Does that mean they're all
Yeah, I'll talk about the ion exchange column one. It's a lot simpler than was talked about in class. I'll get to that in a second. Yes?
So what's the highest rating you can get on exam? Like technically 105?
It can even be higher than that if there's a curve. So there's no limit. You can get 120% maybe. I don't know what it's going to be yet. Mm-hmm.
Wait, it curves?
Yes, it curves each exam.
I read the opposite.
If people do well, the curve isn't very high, but there will always be at minimum a 2-point curve. Yeah.
You just made my defeat.
Yeah. I can't answer that. I don't know what you're talking about. Yes. What's that? 50. questions. Yeah, it'll be like 30, 35 questions, and the extra credit is added after that. So whatever percentage grade you get first, then if you get 5 percentage point extra credit on the extra credit, that gets added after, and then the curve gets added after that. Yeah. It's 50 minutes. 50 minutes. Yeah. 30 to 35. I don't know. I haven't seen the exam. Yeah. Only if no one's using the room afterwards. We don't know.
He said no one's using it.
He said no one's using it? Then that's what we'll do. Yeah. Right. I don't know about extra hour though. I'll have to talk to him about that. He hasn't told me that.
He said he can stay extra.
Okay. I'll talk to him. I don't know that information. All right. Let's see. Before we get to that chromatin exchange, let's see. Oh yeah. This. All right. I answered this in the SI session today. It is recorded. You will be given a style of question where they give you what is called a gel, an electrophoresis gel. And you'd be asked to answer something about that gel. It'll be a long word problem, and they'll tell you there's some disease that is caused by a mutation in a gene. That mutation in the gene, in this example here, it'll be a different example on the exam. The mutation causes alanine to be produced instead of aspartic acid. So the normal healthy person that doesn't have this mutation produces aspartic acid. The one with the disease, with the mutation, produces alanine. What a gel does, what an electrophoresis gel does, is it separates amino acids. Each of these lines you see here represents an amino acid. You don't know which one yet, and we're going to break it down in a second, but each line here represents an amino acid. Each column here represents a theoretical patient. Patient A has these 3 lines in their blood sample. Patient B has these 2 lines, and patient C has those 2 lines. And each line represents a different amino acid depending on where it is on the gel. If a line shows up near the top of a gel, notice it's closer to the positive node. The nodes will be labeled on the gel. So the lines near the top of the gel are going to be what charge you think. If they're closer to the positive charge, what charge do you think the lines represent? Negatively charged amino acids. Your negatively charged amino acids, you should know, and if not, you should study this, are your acidic amino acids. Acidic amino acids are negatively charged at biological pH. What about down here? What therefore do you think the lines at the bottom should represent? Basic amino acids. Therefore, anything in the middle is a mix of polar and nonpolar amino acids. Okay? Yeah, it's a mix of polar and nonpolar amino acids. Okay, so going with that bit of intro, we can figure out a couple things. The person that has the mutation is producing alanine instead of aspartic acid. We just said that any lines at the top must represent acidic amino acids. So patient A must be making aspartic acid, and patient C must be making aspartic acid. But patient B is no longer making aspartic acid from this particular gene. Instead, patient B has this line in the middle here, which based off our process of elimination must represent what amino acid here? Alanine, because it's in the middle, right? Okay, so this person is the one with the disease, whatever the disease is that'll be talked about on the exam. Patient C is completely healthy. Patient C is only making aspartic acid with this line here and is not making alanine. Notice that the basic amino acid doesn't matter in this question because the question doesn't talk about it. The gel has it, but the question doesn't talk about it, so we don't care. So again, patient B has the disease because the middle line here represents alanine. Patient C is completely healthy because they're making aspartic acid because it has a line near the top there. Patient A is a unique situation. Patient A is making both aspartic acid and alanine. For those who have taken genetics, you know that that phrase is called heterozygous or a heterozygous mutation. For those who don't have genetics, another more simpler way of explaining it is that patient A is a carrier of the gene but doesn't actually have the disease. patient A, one copy of the gene is mutated, and therefore he's making alanine, but the other copy of the gene is just fine, he's still making aspartic acid. So patient A is termed as a carrier or heterozygous. Okay, patient B is the only one that technically has the disease. All right, so that's a short explanation of that one. All right, let's see. Now we did, I'll go back to chapter 4 because I forgot this. Cation exchange and anion exchange columns. He probably went into a lot of detail about how these things work, and if you take biochem lab, you actually do need to know how they work. But for the purposes of this exam, all you have to know is these are techniques used to separate amino acids based off their side chain pKa's, or more technically, their isoelectric points. But for the purposes of this question, you can use either one and get the same answer. So what this technique does is, given a mixture of amino acids, figure out what order they will come out of this exchange column. And the way you figure it out is, you will read somewhere, it will talk about what is going on with the pH. I don't care if it's cation or anionic in this case, because they're just blatantly telling me pH is increasing at increments to separate these amino acids from each other. Meaning we're going to start at a low pH, and we're going to end at a high pH. So amino acids that have really low side chain pKa's are going to come out first, and amino acids that have really high side chain pKa's are going to come out last. So that's another way of saying, well, what category of amino acids have very low side chain pKa's? Acidic amino acids. You can go look at a table to confirm that. Acidic amino acids have low side chain pKa. Ones with high side chain pKa, therefore, are going to be what type of amino acids? Basic amino acids. Polars and non-polars are somewhere in the middle. I'll get to that in a second. Yes, question?
So for cation exchange, they could ask the question either pH going up or down?
Technically, its cation exchange will always be pH increasing, but people always forget that over the years, so I just simplify it for them and just say, just look at what's going on with the pH in the problem.
Well, you wouldn't give us like an answer, it's multiple choice with like aspartic acid first and then lysine last and then the two in the middle flip-flop?
It depends on their side chain pKa's. The ones in the middle, and we'll do this example in a second, if you have, for example, like two acidic amino acids, one of them will come out first before the other, and it depends on their side chain pKa in the chart, exactly. So we'll look at this example here. We have two acidic amino acids, aspartic acid and glutamic acid. One of them will come out first. And this is the part where I don't remember what the side chain pKa's are, but you go look at the side chain pKa's of this. I think aspartic acid comes out first, I might be wrong. Whichever one is the lowest one will come out first. So I suggest you guys go check that when you get to that one. The other one therefore comes out next, and then you continue from this. The last category of amino acids that will come out will be basic amino acids. I'm going to skip, and I know lysine is going to be the last one. So the one in the middle must therefore be phenylalanine. Okay? This is the order that it will come out when pH is increasing. If it was an anion exchange column and pH was decreasing, flip everything around. You're going to start from the high end of your pKa table and you end at the low end of your pKa table. Okay? Question. Did you have one?
Oh, I was going to ask about like the anion, so the higher the pKa comes out first. But...
Yeah, so for anion exchange, if that happens to be it, look to see, and it'll tell you it starts at high pH and decreases at increments. Okay. But for cation exchange, it will tell you pH starts low and is increased. All right, so that's how the exchange column stuff works. All right. I think that's it for chapter 4 and 5. That was, again, sorry for it being mixed up a bit. All right. Chapter 6 stuff. So, there isn't actually a lot you need to know from chapter 6 for this exam. Some of the really detailed stuff might show up on the final, but for this exam, it turns out there's not a lot that you need to know. The most important stuff for chapter 6, I covered it in the SI session earlier today, which is also recorded, but I'll reiterate a couple things, are the following bits of information. Know your levels of protein structures. What this means is, back in chapter 4 and 5, you learned that all proteins are made of amino acid sequences. Those sequences, though, by themselves, are just your primary protein structure. If you're just talking about the sequence, you're talking about the primary protein structure. That doesn't do anything, though. An actual protein has a three-dimensional structure. In order to get to that, this amino acid sequence is going to start folding and wrapping around itself to form a three-dimensional shape. It does it in progressive stages. When an amino acid sequence folds and wraps around itself slowly, it first becomes a secondary protein structure. For this class, there's only two types of secondary protein structure you need to know: alpha helices and beta sheets. Of those, there's a couple slides in there that lists identifying properties of these things. For example, and I might not remember all of them, so you got to go look in the notes for this. Alpha helices are right-handed helices, meaning they